In this video, we will discuss about this application of this HEMICA microfluidic device to analyze MDA-MB-231 cells. As these MDA-MB-231 cells informations are already known, so the results of this HEMICA microfluidic device will help us to know how accurate how reliable this microfluidic device is by comparing the results from this experiment with the results that are already published in the literature about this MDA-MB-231 cells. So we have already discussed in our previous videos how to fabricate this HEMICA microfluidic device and make a gel out of this microfluidic device. So let us start with this, and let us start with the cell migration speed. And one more thing that we need to know before we go into the analysis is that these experiments are done on a confined region. That is, the width of this microfluidic device is maintained at across 4 micrometer. So in the case of this cell migration speed, we can see that the migration speed is high when the apicolateral gel has a high stiffness, that is a 15 kilopascal. That they have used two different stiffness of the gels, 15 kilopascal and 8 kilopascal. So when the apicolateral or upper gel has the high stiffness around 15 kilopascal, we can see the high migration speed. And when they check the persistence, we see the similar thing, that is the gels that have high stiffness at the apicolateral has the high stiffness, with the maximum stiffness achieved with has the gels which have the apicolateral has the gels whose stiffness is high at the apicolateral, or we can say that gels with high stiffness at apicolateral layer has the high persistence, and with the maximum persistence observed for both the apicolateral and basal gels has the high stiffness. Later on in the panel C, they have checked this chemotactic gradient, whether this HEMICA microfluidic device can be used to measure cell migration in the chemotactic gradient or not. And they can see that the movement of the cells from one region of interest to the another region of interest, when they have used a different kind of media, that is, one media contain FBS and another media does not contain FBS. So they can see the chemotaxic effect in the case of in that case, when they use this HEMICA microfluidic device, providing info or substantiating the fact that this HEMICA microfluidic device is capable of measuring this chemotactic gradient effect, such as this cancerous cells. When cancerous cells are more prone to acquire the mesochemical properties under the effect of TGF beta, so that those cell migrations effect can be monitored using this HEMICA microfluidic device. Later on, when they quantify this movement of the cells under this chemotactic gradient, they can find out that there is almost all this movement at all the regions are almost similar, with the highest movement is observed at the upper region of interest, and the slight lowest movement is observed at the lower region of interest. But however, if we look at in general, all the speed velocity after a certain time, that is around 10 to 15 hours, is almost same for all the region of interest, either that is upper region of interest or lower region of interest or this macrochannel region of interest. And thereafter they quantified the number of cells that are successfully transferred from one region of interest to another region of interest. And they can see that a maximum number of cells are transferred from lower region. As they seed the cells at this lower region of interest, they can see the maximum number of cells are transferred to the upper region of interest, and some of the cells remain in the lower region of interest, and some are uh, found in this microfluidic channel. And the speed rate when they check the speed rate when they check when comparing the case when there is no difference in the no difference in the media compared to the case where there is a difference in the media. There is one media contain 10% FBS and one media does not contain any FBS. They can see that whenever there is a gradient of 10% FBS, they can see the high speed rate. So this further substantiate their point that this device is capable of measuring this chemotactic gradient. And when they check the persistence of the cells, they can find that there is a increase in the persistence when the cells has a gradient of when the system has a gradient of 10% FBS. And thereafter what they did is they check whether the cell movement are in the form of bulk or in the form of single. So in this case they use different kind of width to check whether to check the cell movement or cell motility. And they can see that when the width is high or when the movement is like 2D like movement, they can see the cells are multiple cells are moved. Multiple cells will be able to move parallelly or at the same time. However, when they see that when the channel width is decreases, they can see that this multiple cell movement growth rate get reduced, and it will acquire more and more the single cell movement that we can see in this J panel. And I is the transition form, where we can see some of the cells are in some of the cells move together, whereas as the width of this microfluidic device decreases, we can see the more and more single cell movement. So this provides us the information about how this width can be used to control and to customize with based on the experiment need, whether one needs to check the single cell movement or the whole cell or high throughput cell movement. So this overall this figure provides the information about how this chemotactic gradient effect can be observed in this HEMICA microfluidic device, and the effect of channel width, or in other terms we can say we can measure either single cell movement or the 3D cells or high throughput cells migration in this HEMICA microfluidic device by varying the width of this HEMICA microfluidic device. And if you need to understand more about this HEMICA microfluidic device and its application, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.